So this is an A-level biology presentation looking at populations. So when talking about ecology and populations, there's a number of terminologies that you need to be absolutely solid on before you go any further. A number of these you may have encountered at GCSE, but if you have forgot them, you need to make sure to memorize them. So pause the video at this point and go through each of the statements in front of you and see what definitions you can come up with. When you've done your best, restart the presentation and look at the definitions. Check off your answers, correct your answers, and I suggest that you memorize the definitions that you've been given before you progress any further through the presentation. So pause now and see what answers you can come up with. When you're ready, restart for the answers. So these are the definitions that you should memorize before you go any further through the course. As I say, a number of them are GCSE, but just make sure you're 100% before you go any further. So there are several isolation methods that ensure that you have reproductive isolation or separation of organisms, which means that they will be classified as different species. Again, folks, pause the video at this point and see if you can work out what the 7 pictures are. And when you're ready, go through each of the pictures and you'll get the type of isolation mechanism. So pause the video now. In terms of isolation methods, what you have is you have ecological, geographical, mechanical, behavioral, temporal. Again, it can be seasons, but it can be times of day and any kind of time whatsoever. We have gametic and we have hybrid sterility, where an organism is produced, but that organism is sterile. As part of this lesson, you're going to look through a number of different terminologies, which you've already done at the start of the presentation. We're also going to have a look at carrying capacities, biotic and abiotic factors, how to measure biotic and abiotic factors, as well as the concept of niche. So to start moving through, we've got biotic and abiotic factors. So biotic factors are living factors. The abiotic factors are non-living factors. In front of you, you have 11 pictures. Pause the presentation, see if you can work out what each 11 pictures represent in terms of biotic and abiotic factors. When you've done your best, you can restart the presentation and we'll go through the answers. So pause the presentation now. So if we take each one in order, we start with the left-hand side, number 1. We're going to look at the abiotic factors. Again, folks, you got to be careful in your terminology. If you look at that picture and says something like sunlight, that's not enough information. We actually need to talk about intensity. So it'll be sunlight intensity. We have the volume of precipitation. Again, not rainfall, not precipitation. We have to talk about volume. Soil pH. Again, just putting pH in there is not enough detail. You got to be specific. We've got temperature. We have humidity. And the last one, we're talking about carbon dioxide concentration or CO2 concentration. Again, make sure if you're going to use the chemical formula CO2, make sure you use a subscript 2 that goes well below the line on any answer. On the right hand side, we have biotic factors. And what we've got is photosynthetic organisms. That does include all your plants, but obviously algae and anything else that will photosynthesize. We have our herbivores and our carnivores. We also have what are called decomposers, and of course our cells, which are the omnivores. Make sure you can identify any one of these. Could be talked about in an exam question. They will give you data and they will ask you about how it affects different organisms, so make sure you are aware of it. There's an extension question which asks about which factors have the biggest impact, biotic or abiotic. If you have not thought about that, pause it now, spend a minute, see what you can come up with, and then restart the video. So pause the video now. In terms of which has the bigger impact, well, there's no real way of measuring which one will have the bigger impact. There's arguments for both. You could talk about the fact that the abiotic provide the habitat and as a consequence will impact on what biotic organisms are there, and therefore they're more important, and vice versa. If there's no biotic organisms, well, then they're not going to affect the abiotic organisms. So it could go either way, but just be aware that these things are very much interconnected. In terms of how do we measure them, so for each one we have to come up with a method of measuring that particular variable. So you've got the same list on the left hand side, exactly the same as the previous bit of the presentation where you had the pictures, and all you've got to do is identify how you can measure them. Most of the time we use specific bits of equipment, but there are a range of methods that also can come into play. Pause the presentation at this point and see how many you can answer. So we'll go through the answers. For sunlight intensity, we literally go with a light intensity meter. A photometer is the actual term for the piece of equipment, but you would get away with light intensity meter in the exam if you're really stuck. But photometer is the best one. In terms of volume of precipitation, we just need anything that will measure volume, such as a measuring cylinder. For soil pH, we use a pH meter. Temperature, I hope everyone would get, is a thermometer. Humidity is what we call a hygrometer. CO2 concentration is a CO2 meter. You might start to see a pattern at this point. Photosynthetic organisms, we normally measure area using a quadrat. Obviously, if we are talking about some sort of aquatic photosynthetic organism, so algae in a pond, quadrats become a little bit more tricky and we have to find some other way of measuring the area. For herbivores, we can use capture release method. For decomposers, we normally use what's called a pooter. We've also got mark capture release for carnivores and for omnivores. You can even go in and use different methods of capturing organisms. So for example, pitfall traps, but it's normally capture mark release is the method. So we use this term niche, and it's kind of a tricky one to define and people get it very easily confused. But a niche is essentially an organism's role in an ecosystem. It's not just where it lives, it's everything connected about it, all the biotic and abiotic factors that are in some way connected to that organism. When we go talk about niche, we have two types of niches. We can talk about a fundamental niche, or we can talk about a realized niche. A fundamental niche is the potential niche of the organism, everything that it could be connected to. The realized niche is what's actually connected to. So for example, a fundamental niche may be different organisms that could be prey for a particular species, whereas the realized niche are the actual organisms and species that it's eating. With a range of niches, every organism has its own niche, and because it has its own niche, they're not competing for the same material, and less competition means that those organisms are more likely to survive. If every organism had the same niche, they'd all be competing for the same resources, and as a consequence, many of them would not survive. This is an example of the kind of question you would get on niches in the exam. It's given you a rather complex little diagram talking about different factors, in this case temperature and size of food item, and it's asking you to interpret it. And what they've given you is what we call a bell-shaped curve, where if you look at species A and species B along the bottom where it says size of food item, species A and species B simply have a bell-shaped distribution where you have the highest point is where you have the largest number of those particular organisms. And you can see here that this line represents all of where we find species A. And up here, if we look again, you can see that anywhere along here we have species A, which means that this grid 1 represents where species A would live. If species A live there, that is their niche area. That's where they're more likely to be found. Again, species B, if we look at species B, you can see that they have area 3. The next question says, insects too small for species B and temperature too warm for species A. So we're looking at temperature, we're looking at where species A are, and we're using the left hand side. Here's temperature. We're looking at a higher temperature at the bottom, and we're looking at here and 2 and at 4, we have got no species A. We also have no species A on area 3, but we'll come back to that in a second. So 4 and 2, we do not have any species A. We also do not have any species B, which is why the answer has to be 2 or 4. In terms of species B, we're talking about insects being too small, so we're looking at the size of the food item. We're looking at the smaller end of the size of the food item, which means we must be talking about area 4. Where we've got insects too large for species A, well, again, we're talking about size of food item, and we're talking this time about the larger end of the size of the food item, which is where the arrow is on the right hand side. And we're talking about a temperature, but we're looking at species B, and we're looking at the lower end of the temperatures, which means we're looking at this end of that arrow, which is why we are looking at area number 2 for the last section. We do have an area that isn't discussed in the question, which is area 5, which is suitable for both species A and species B, and is the location where we would have the highest level of competition between species A and species B. And just to finish off, make a list of all the words that have been covered during this presentation. Then go through and see how many you can actually draw connections between and put some sort of connection between them. Go back then, watch the presentation again, change pen, add in extra words that you missed the first time round, and then try and build even more connections to try and get those other words to stick into your head.